Um, okay, so I'd also like to welcome the participants in the course in Limoges, and we're really glad that you made it to Limoges, and hopefully you have a good time there. And I'm also very, very thankful to the local organizers. I can imagine how much work it is to organize everything and get things smoothly done. So again, Veronique and your team, really a great thank you. They asked me to give a short overview of BovReg, are we doing in the project, and maybe also what is the connection to this ATAC-seq workshop. BovReg is a new project on the identification of functionally active genomic regions relevant to the diversity and plasticity in cattle. BovReg is part of the functional annotation of animal genomes FANG, and I think Emily will probably give some more details on this. So Emily, can you respond? You will probably introduce a little bit more about FANG. Emily? Okay, so just in a nutshell, FANG and its new counterpart, or its new subset, Eurofang, they are European and a worldwide initiative for establishing open access resources to help functional annotation in farmed animals. Global FANG has now more than 500 partners, and in Eurofang, this is kind of an umbrella for several recently or some time ago launched EU projects, including BovReg. Members are also GeneSwitch, AquaFang, which you already know, and some very recently started projects like Rumigent and Geronimo.
Christa, excuse me. Can you put on a presentation mode, because the slides are not running?
It is a presentation mode.
No.
That's strange, because do you see it now in presentation mode?
No.
Do you see it now in presentation mode? I have a very slow. I try again. The terrible echo.
There's someone connected, someone go or not? Your computer?
Right?
Yeah.
Yeah, that's fine now, Christa.
Okay, again with the echo. So it's better now with the sound.
Let's try.
Is the sound better now?
I think so. Yeah.
Yes.
Yeah.
Yes.
Okay, so then BovReg is an EU-funded project with a focus on biology-informed breeding, so to assist the next step from moving away from anonymous S&P information to more biologically supported weighting of particular variants in the genome. all in all, our aim is to generate and validate functional genome data. And we also want to have a standardized technologies for data analysis. We want to do this for, there's a lot of background noise now for me. Okay. Yeah, okay. Because I hear very, very substantial background noise. Okay, I try to ignore this. So it's based on, BovReg works based on Func core assays, and with this we want to provide a catalog of functionally active genomic features, and we will do this for the Dummerstorf genome, for all of those who are working mostly on Bos indicus, and I remember there was one colleague from Queensland, and maybe that there the focus is Bos indicus. So we're really focusing on Dummerstorf. Nevertheless, Bos indicus should not be that different, but because we are a European project, we did not include Bos indicus, at least not specifically. Our key traits are robustness, and for this we will look at fertility, health, and that's represented in our project by mastitis susceptibility, and we also get biological efficiency, and there we have a look into feed efficiency. The timeline originally was 4 years, but due to Corona, we recently had an extension of the project, so it will run until February 2024. Specific objectives are that we decided we want to have new laboratory tools and bioinformatic tools. We want to annotate the functionally active genomic regions in those tissues that we think are really very relevant to our target traits. And we want to have a wide distribution of tissues and animals to cover as much as possible the whole variety of functional active genomic elements in the bovine genome. So we also have different genders and also different autogenetic stages. Furthermore, we also look at some cell lines and provide resources for those. Besides the normal traits, which zootechnical traits like growth or milk production or health, we also use new molecular phenotypes like transcriptome or the metabolome to further map those features to the new features to those regions which are functionally relevant. And with this knowledge, we will develop this knowledge feed into prototype models for improved genomic selection. So in a way, we would like to prioritize genetic variants with respect to their annotation with, in terms of functional active regions or functional active motives. Besides that, we also provide training, dissemination, and communication activities and have a view on social perception. And training, you also are just part of this activity. We have a dedicated training session not just on wet lab, but also on using the information we generate in BovReg for genomic selection. BovReg has 20 partners, 19 funded partners, and 1 partner from Australia, not from Queensland, but from Victoria. We have represented 13 different countries and also have a member in BovReg from Canada. All in all, we have 11 different work packages who interact with each other. And can you see my mouse?
No.
No.
No? Okay.
And we see the slide 5.
The slide 5. Slide, okay. But this slide 5 is with a different, is the map.
Yeah.
And you now see another. Now you see the different work packages?
No.
No.
That's strange. Now you see something else?
No.
No.
Still
Is
the
that
slide
still the
5.
slide?
Yeah, slide 5.
Okay, but I can't do anything about that. That's weird. Can you see now the open?
Now it's changing.
Slide 6.
No. Yes.
Now we see the work
And now?
packages.
Okay. Let's see whether it's moving forward. And can you see the laser pointer now?
No. No.
No laser pointer. Okay. So we have the different work packages with the tools and resources and bioinformatic tools and pipelines on the bottom, which are applied then to three other work packages which generate data, which then feed into a data portal. But from that, there the data will be used for biology-driven selection. It will also be then forwarded to a training, communication, dissemination to be useful for the industry, for the public policy, and also for the scientific community. To have this a little bit more fed by specific data, because this is still very abstract, we all these work packages into these 6 activities, which is establishing laboratory and bioinformatic tools, the comprehensive genome annotation map per se, then data integration for biology-informed breeding, to distribute the data, to outreach and social and societal impact, and then also have activities integrating the different FANG efforts. So this established laboratory and bioinformatic tools. So we are developing laboratory tools and resources. For example, we will looking at cell lines, which are very, very frequently used in not just neuroscience, but also veterinary science. And as a very brief example, you can see here kidney and the MDBK cell line, which is supposed to be a kidney cell line, and you can already see that this cell line very substantially differs from its mother tissue, highlighting the limitation of pure cell line work. Then we have the tools, data, and pipelines for bioinformatic analysis, and I'm quite confident that you will hear a lot about this from Jose and his colleagues, because we agreed together with the other Eurofund projects to implement standardized and harmonized pipelines for reproducible data analysis, all based on Nextdoor core, and we set up a GitHub repository with information on, for example, workshops, but also on the new developed pipeline data analysis.
Sorry,
on the other hand,
Christa, sorry.
Yeah.
On what slide are you? Because we can see only nothing is moving. And
Slide 8. Slide 8.
Okay. Okay, it's good.
No, but
No.
yeah, it's
now.
fine now. But
Sorry.
Now.
Yeah, it's now 8.
yeah.
Yes.
It's 8.
You are not in presentation mode.
It's moving.
Yeah.
It's not on the presentation mode. It's moving one by one.
Because on my screen it's presentation mode. Maybe really the best thing is that I try to make these things as possible.
Yeah. Yes.
And enlarge as much as possible.
Yes. Yes. Good.
It's not in presentation mode, but then at least we are
on the same track. Yes.
You will on the same slide then, yeah? Okay. So this is about the pipelines. The portal where all the data from BovReg are hosted. It's the FANG Data Coordination Center, and their portal is developed which hosts the data, but also activities were conducted to enable feeding these data into the portal. And also new features like, for example, livestock ontologies are implemented there. So you might know that when you want to do pathway enrichment analysis, for example, or if you want to reuse data, ontologies are very important. But we were aware that the status of ontologies with particular features for our livestock animals is rather poor. For example, anything related to rumination is probably not very well covered in annotations, mostly based on humans and mice. Or I saw 2 colleagues from aquaculture. You probably also suffer from very poor annotations sometimes. So we have the Data Coordination Center at the hosted EBI, and the unique feature of FANG and also of BovReg is that we don't just deposit assay data as you might be used from open access publications where you have to deposit your assay data, but also our data are associated with very, very extensive metadata. So from the samples which are used to generate those data. You can have a lot of information about which kind of animal do these data come from, when is it born, when were the samples taken, how were the samples taken, and all the other aspects which can have a substantial impact on later outcome of the assays. And finally, also the methods that come with the assay will be distributed so that there are whole packages. I mentioned we want to build resources for functional annotation of farmed animals, and so not just the data itself, but the data and also how the data were analyzed come as one package in the FANG data portal. And it's also linked to the INE and biosamples archive, so that there's no triple data deposition, but it's all integrated into a concept of reproducible and findable data. So coming to the annotation map per se, because this is the core part and also I think the part which is next to this workshop, we have the functional and structural annotation map. So we have different assays, and many of these assays, they are all applied to the same samples, which is something which is, I think, to some extent rather unique in BovReg. So we have the transcriptome, chromatin excess, interacting regulatory regions, and also mobile genomic element insertion sites. To give you a flavor of this, Gabriel, who attends this workshop from Liège. The team in Liège, they did very, very extensive surveys across 6 months, which are a core part of BovReg, from Holsteins, from a beef cattle population, and from crossbreds. They are neonates, they are juveniles, they are adults, and all in all, about 130 tissues from different environments. And across these different tissues and samples, we working with short read sequencing, library preparation, and we are covering the entire range of transcriptomics from mRNA-seq to CAGE, including total RNA. We will have also ATACs, so that's probably what Gabriel is going to talk a little bit about. We are doing ChIP-seq assays for 5 chip marks, and all these data, they are analyzed in a standardized way with Nextflow and NF-core based pipelines, which are accessible, and so all the data are which are generated from the raw data to the data analysis per se is reproducible and useful for later analysis. So this covers mostly the core assays. And then what we also do to foster the functional and structural annotation map is we are doing epigenetic studies, mostly on methylation, to find specific environmental or inter- and transgenerational effects. So for this, we using bisulfite sequencing as well as an enzymatic method for methylation sequencing, and establishing methylation patterns on the one hand on embryos exposed to different environmental stresses, including heat stress. Within specific experimental designs, we are monitoring also intergenerational and transgenerational effects, and also will highlight or are highlighting regions of the genome which are particularly sensitive to these environmental effects and also to epigenetic modifications. And as the final layer of annotation, we are doing eQTL, MQTL, and MetaGWAS. So this is all about functional active regions in terms of QTL on the one hand, so technical QTL like the ones you probably already know, like P deficiency or mastitis incidence or milk yield. On the other hand, we also have metabolome analysis and do metabolite QTL and metabolite MetaGWAS. And finally, we're also doing expression QTL studies comprising quite a substantial collection of RNA-seq samples. So when you put on top of each other all the different layers, you will see that some regions really seem to have a rather high impact. You can see this little red dot here. Obviously, if you put on top of each other, this seems to be a variant which has a very, very high likelihood that indeed something is going on here. The variant in this region, at this particular position, we could expect that because the region is transcribed, because it shows that there are it's interacting with other regions, it's sensitive to methylation, it has an epigenetic response, as a signal from MQTL, EQTL, metaGBA. So indeed, these dots here, these positions have a really high impact on the diversity of cattle. And as a exemplarily, some of these variants will be validated not just by these association studies, which more or less is just an indirect evidence, but we're also doing CRISPR and reporter gene assays and promoter interaction assays to really validate that these predicted regulatory variants are truly functional. But all in all, these maps, they will feed into mostly biology-driven genomic prediction models, which is at the moment, many genomic prediction models treat all the different variants equally. So there is no prioritization. And the question is how to best do this prioritization. So a variant like the red one should have a definitely higher impact on the prediction model than, let's say, a variant which is here where there's not that much of interest. So our colleagues from quantitative genetics, they developed such models, how to best make use of this information from functional annotation to improve genomic prediction. What we also do in BovReg is so sometimes people from society, they claim that scientists just do their work and find new methods, but the acceptance of those methods is rather poor in society. So we already, in a way of preparatory and in advance, looking for potential impacts of our new technologies, we have a work package which explicitly deals with ethics and societal issues. So we want to engage the public and see what is the social perception of the new breeding technologies, including genomic prediction. And this is also for stakeholder organizations to synthesize these analysis into frameworks to help them to address also the public. And finally, we are doing efforts on training, communication, and dissemination. This is to interact with the online projects. We are conducting wet lab, bioinformatics, and statistical modeling courses. So the first two parts you already have in this ATAC workshop. We do a communication about the project, go for conferences, demonstration. And I think I keep it at this. So this is again the last team of 20 different institutions. And from time to time we meet. And I'd like to thank you for your attention. And I hope that you will also have fun with the workshop like the team had here at our kickoff meeting with BovReg. And I think I kept this short to give also the participants the chance to ask questions about BovReg, things which I might not have touched or where you might have specific questions to BovReg.
Thanks, Christa. Maybe we have time for a few questions.
I don't know if any of you here in Limoges have some questions to ask Christa about the BovReg project. No?
Online too.
Yeah, and people online also. In the chat box. Currently, looks like in the chat box there is no questions so far. Okay, so if there is no questions, because Christa was very clear, you know, so it's good. Thank you, Christa. So I don't know, Leonie.